In this lecture, I will discuss about TH1 and TH2 cells, how these two cells are generated and what are the effector functions. At first, I want to discuss about TH1 cells, how these TH1 cells actually generated. So for the generation of TH1 cells, three cells are very important. That is dendritic cell, macrophage and NK cell. TH1 cell is generated in response of some specific bacteria and protozoa that can survive in the phagosome. What happens? The dendritic cell macrophage can phagocytose bacteria or protozoa. Okay, but some bacteria and protozoa have evolved some mechanism that they can survive and replicate in the phagosome. Example is Listeria monocytogenes, mycobacteria like Mycobacterium tuberculosis, Mycobacterium leprae, and some protozoa like Leishmania. So this pathogen can survive inside the phagosome. So in response to this pathogen, this dendritic cell, macrophage and NK cell can secrete some chemicals called cytokine. Like this dendritic cell can secrete IL-12, IL-18, this macrophage can secrete IL-12 and this NK cell can secrete IFN-gamma. So there are three cytokines that act on T cells. T cells. Let's assume these are T cells. T helper cell. At first, the T helper cell is naive in condition. Naive means it is inactive in conditions. But the dendritic cells can activate in naive T helper cells. So first thing is activation of naive T helper cells. I have made a separate video on T cell activations. I am not mentioning here the activation of T cells, but I am telling you that the naive cell must become activated. So it become TH effector cells. Now what happens after when this dendritic cell, macrophage and NK cell secrete these cytokines, they are received, this T helper cell actually receive this cytokine by cytokine receptor. So they can receive this cytokine, and in response to this cytokine, this T helper cell actually produce a molecule called T-beta. This is the master regulator for the TH1 cells. When this T-beta transcription factor is produced, the cells become TH1 cell. Okay, now this TH1 cell can secrete chemical like cytokine. Specifically, I am mentioning IFN-gamma. This IFN-gamma actually amplify the TH1 production, because you can see here the NK cell is producing IFN-gamma that is actually inducing the generation of TH1. And also this TH1 is producing IFN-gamma that actually helps in the amplification of this TH1 cell. And another thing is important, very important, I want to mention here. This TH1 cell, when this TH1 cell is generated, they actually inhibit TH2 cell generation, they inhibit TH17 cell generation. And this IFN-gamma can act, can act on B cell. The B cell receive a signal to produce antibody, like IgG3, IgG2A. These two antibodies actually helps in complement activation and opsonization. This IFN-gamma can act on also macrophage, because macrophage have receptor for IFN-gamma, and when IFN-gamma is received by this macrophage, a very important thing occurs, that is this macrophage starts to produce, transcribe and translate some proteins called INOS, ROS, reactive oxygen species, and this INOS means inducible nitric oxide synthase. Inducible nitric oxide synthase. This enzyme can produce nitric oxide that is actually reactive nitrogen species. So what happens when IFN-gamma act on macrophage? The macrophage actually gets activated. It starts to secrete or produce very important some molecules that actually digest the intracellular pathogen. Let's assume this is the intracellular pathogen, bacteria that is present inside the phagosome. So this is actually the effector function of TH1 cells. Okay, so TH1 cells is generated in response of intracellular bacteria that have actually evolved some mechanism to survive in the phagosome, and after production of TH1 they can induce B cell macrophage. Okay, now I will tell you the TH2 cell generations. TH2 cell is generated in response of different microorganisms. This is actually helminth. So let's assume it is a helminth. TH2 cells activated in response of helminth infections. If it is a helminth, what happens? This helminth have chitin and some other cells, other immune cells like mast cell, basophil cells. These cells can sense the chitin of this helminth. So it is a helminth, and in response to this chitin or this helminth, this mast cell and basophil cell can secrete some chemical called IL-4, and in response to this cytokine, any naive TH cells can become TH2 cells. So this is very important. For the generation of TH2 cells, IL-4 is just enough. So this IL-4 is received by this TH2 cell. This is a receptor and it recognizes the IL-4 and become TH2 cell. Okay. Now this TH2 cell can secrete some cytokines. And what happens? Actually IL-4 activate a master regulator in case of TH2, that is called GATA3. Not only that, this TH2 cell also can inhibit TH1 and TH17. So this cell can inhibit other cell generation, and this cell, TH2 cells, can produce 3 cytokines. One is IL-4, another is IL-5, and another is IL-13. I will tell you the function of different of these cytokines. The first thing you can see that this cells is producing IL-4, and the cell actually TH2 cell is generating or is actually induced by the IL-4. So this IL-4 actually helps in amplifications of these cells. So amplifications. Another important function of IL-4. In response of IL-4, B cell, okay, this is B cell. In response to this IL-4, B cells have receptor that can receive this IL-4. It secretes a specific type of antibody, that is IgE. I will tell you the significance of this IgE. This IL-5 actually activate another cell called eosinophil, and this IL-13 actually helps in production of mucus by the goblet cell and peristalsis. What happens? Mast cell, basophil cells and eosinophil cell. These 3 cells express a receptor called Fc epsilon receptor, and this receptor can bind with the Ig molecule, Fc region of the Ig molecule. When, let's assume this is a mast cells, I am showing you with this mast cell exam. So these mast cells have a receptor that can bind with IgE, and all these cells have this receptor. Let's assume this looks like this Fc epsilon R, Fc epsilon R. This can bind with Ig antibody. So Fc region of Ig antibody, this is the Fc region of this Ig antibody, this region, it is Ig antibody. What happens when these 3 cells, mast cell, basophil and eosinophil, bind with this IgE antibody? They secrete, actually degranulate some granules. There are granules present in this mast cell, basophil and eosinophil. They actually secrete the chemicals from these granules. This process is called degranulation, and this chemical actually kill this helminth. This TH1 and TH2 cell can cross regulate each other. How? I am telling you. TH1 cell is generated in response of IL-12. When IL-12 is received by helper cell, it starts to express a master regulator that is called TBET. Master regulator TBET, and this TBET can help in the production of, positively induce production of IFN-gamma. Okay, and let's assume in case of, so this is the TH1 case, TH1. In case of TH2 cells, the IL-4 cytokine is responsible for induction of a master regulator, that is GATA3. Now what happens? This GATA3 actually helps in the production of, positively regulate production of IL-4, IL-5, IL-13. Now what happens, this TBET and GATA3 inhibit each other. So if one cell, let's assume this is a TH1 cell, IL-12 is received, then TBET is produced. This TBET actually inhibit GATA3 molecule in that particular cell. And if it is TH2 cell, then IL-4 actually received and GATA3 is produced. GATA3 actually inhibit the TBET. So once a cell become TH1 or TH2, it cannot become the other. TH1 cell cannot become TH2 because the TBET inhibit GATA3 and this GATA3 inhibit TBET. Not only that, this TBET actually inhibit IL-4, IL-5 and IL-13 productions, inhibit, negatively regulate, and this GATA3 also negatively regulate production of IFN-gamma. So you can see this TH1 and TH2 cells actually cross-regulate each other. There is another important function I want to mention here. The another important function performed by these TH1 and TH2 cells is activation of macrophage. Let's assume this is a monocyte. What happens when a macrophage is inactive in form? It is called monocyte. This monocyte that actually become macrophage can be activated by two ways. If this monocyte is activated by TH1 cell that is secreting IFN-gamma, then this activation is called classical activation, and the macrophage that is produced in response of this TH1 is called M1 type macrophage. This M1 macrophage actually induce ROS production, reactive oxygen species, reactive nitrogen species, some lysozyme enzyme and some cytokines like IL-1, IL-6, IL-23 and chemokines. All these actually helps in the killing of this bacteria. So this ROS means reactive oxygen species, reactive nitrogen species, kill the phagocyte cells, kill the bacteria or pathogen, and this chemical actually helps in production of inflammations. So it helps in inflammation, so that many immune cells come into the infected site. Inflammation is a red signal, so that many immune cells like neutrophils, they come into that site to help this macrophage in the killing of this pathogen. If this is the TH2 cell, it can activate this monocyte but in a different way. These TH2 cells actually secrete IL-4 and IL-13 that actually helps in this macrophage monocyte to transform into another type of macrophage that is called M2 type, and this activation process is called alternative activations. M2 macrophage secrete different cytokines like IL-10 and TGF-beta that are actually anti-inflammatory. This is not inflammatory, this is anti-inflammatory. This macrophage can inhibit the inflammation